So I did. I put a short disclaimer here from Carl Linnaeus, which I thought was kind of funny. If you let me go back here. There we go. Yeah, he says if you have remarked errors in me, your superior wisdom must pardon them. Who errs not while perambulating the domain of nature, which of course appeals to all of us. I was going to start off today by showing you a bit of taxonomy here. And where ferns actually fit in the plant kingdom. And of course, this may be repeat or review for a number of you, but if you look at all the vascular plants, which are called the Tracheophyta, there's a division between ferns and the seed plants, which is a major category or two categories in the kingdom. Ferns actually get divided into two subclasses, the eusporangiate and the leptosporangiate, and this really just has to do with how the plant produces its sporangia and basically exhibits them on the fronds of the fern. And then, there's some dispute here, but, whisk ferns, grape ferns, horsetails are usually included in this fern and fern allies collection. And then you can see here the Polypodiopsida is a series of subdivisions all the way down to the Polypodiales, which is where Staghorn ferns reside. And looking from left to right, there's an assumption by plant taxonomists that the farther out you go into this tree, the more advanced and more recent in time is the interpretation. If we focus on Carolina, I looked at two references for us. One, the Manual of Vascular Flora of the Carolinas shows that from a native fern standpoint, there's 16 families, 30 genera, and 89 species. And then comparing the other view, if you check out the UNC Herbarium website, they show that there's 22 families, 46 genera, and 122 species. And I think that may be because the Manual of Flora is basically a key, and then the herbarium, of course, is showing both what is native and what is invasive. So that may account for the reason of the higher numbers on the herbarium website. You'll notice here the 16 families I've listed that are in the vascular flora of the Carolinas. And then I'm showing you here the common names, which hopefully resonate with you. And I'm sure many of you probably have these in your collections. Again, I'm not sure that the Selaginellas are, really true ferns, but they, and again, Psilotum or whisk fern is also included here. But again, when you're studying these things in school, botany departments typically, talk about fern and fern allies. And these are basically vascular plants that do not bear seeds. So how does since we're talking about propagation from spores, where do the spores come from? And this is a graphic that shows the life cycle of a typical fern plant. And the key aspect of fern reproduction really has to do with this alternation of generations. And you'll see here, these particular features of the fern life cycle are actually haploid, which means they only have half of the complement of genes in every nucleus in every cell. So from the spores through the young gametophyte with the sex organs, these are all haploid cells within these aspects of the fern's presence in nature. And then the surprising thing, I think, to a lot of people, when you think about plants, ferns and fern allies actually have motile sperm cells, just like animals. And in nature, spores typically are flying around in the air when the plants are bearing them. They settle, in favorable habitats like, mosses or liverworts, or any moist area is going to allow the spore to germinate and produce this very primitive collection of cells called a gametophyte. And the gametophyte actually produces the sex organs, which are external to the little plantlet. And again, if you think about seed plants, all of this is actually all of these features are actually contained in the seed, whereas in a fern, they're actually external to this little plantlet. And then, if you think about the gametophyte growing on either, a wet tree trunk or, on a moss outcrop, when it's raining or just after a rain, the antheridia, which is where the sperm cells are created, will pop open, and the little sperm cells will swim around looking for this feature called an archegonium. And inside the archegonium is where you have the creation of the egg cell, for the new fern plant. And again, these features are all haploid. When fertilization occurs and you result in this zygote, you actually have now the combination of two complements of chromosomes. And so all of these features now in the life cycle have a full complement of chromosomes. The gametophyte will produce a little plantlet, which actually matures into the plant that we know, by its common name. For example, this might be a Boston fern. And as the Boston fern, for example, matures, you'll see these little dots on the back of the leaflets of the frond. And there is what's called a structure called a sorus, and the sorus actually is a little umbrella that protects the sporangium or the sporangia from the weather. And as the sporangium matures, it will then cross this line and produce more spores. And that's the essentially the leaf cycle of a typical fern. If you want to culture ferns from spores, there's a variety of methods. And this is actually the method that I used in grad school, which I essentially made up because I really didn't have a guiding light at the time. Certainly I had my major professor coaching me in terms of what to try, but now there are several containers that I'm showing here. And if you dust spores onto moist sand and hold them, at a fairly warm temperature, 75 to 80 degrees Fahrenheit, for example, using your seedling heater, they will germinate in about a week. You won't be able to see them. They're microscopic. But if you're patient and, watchful, you'll see the gametophytes begin to appear as little green dots on the sand. And just for reference, a typical fern spore, at least in terms of the staghorns, is about 35 microns long, which is probably around 1,000th of an inch. So, when you collect spores, you can see them as sort of golden looking dust. But individually, they're single cells that are microscopic. In grad school, I used a very dilute solution of rapid grow, and also I was able to make up a media that one of my other professors was using to grow algae in the laboratory, and that actually turned out to be a really good media as well. Here are some photos to show you essentially a couple of individual fern spores. And, as the conditions in your culture dish become favorable, the spores will crack open and begin to exude this little filament. The filament cells began to develop these unicellular rhizomes, which function, in microscopic land as roots for the emerging gametophyte. And this actually is a series from Platycerium coronarium, which I'll show you later in terms of where it's native. But, my point here is collecting spores from nature is easy. You can certainly collect them from your garden. If you scrape them off into a small container with a lid, keep them cool until you're ready to sow them, and then dust them onto your moist culture media using an artist brush. And I'll show you the reason why you would use an artist brush as the applicator. Once exposed to water again, the live spores, which are single cells, begin growing by producing a very small filament. And as you can see here, this filament actually produces chloroplasts, and chlorophyll is present so that the little creature can actually make its own food. As the fern gametophyte matures, you can see there's a formation of a little spatula-like plate that begins to divide. This little plantlet only has probably less than 20 cells, but as it matures, it will grow into a very small, flat what's called the thallus, which has hundreds of cells. And these pictures are Platycerium coronarium and Platycerium grandi. So the series shows you that the germinated spores develop into the gametophyte, which then begins to develop the sex organs containing the egg cells and the sperm cells of the mature plant. And you, again, with patients, will need to wait 4 to 6 weeks for the gametophytes to mature and begin producing these external organs. Since these are tropical, I'm also recommending that you use a 12 on, 12 off light cycle for these plants, because that's what they would see in nature. Here's a couple of photos that I made. This first one was made with a phase contrast microscope at about 20x, which is a very low magnification, but this gametophyte is about 5 weeks old, and you can still see the little spore capsule here where the plant, our plantlet begin to emerge. And these structures are the rhizomes that allow this thallus to absorb water and basically sustain it. This is another photo, and I apologize for the photography here, because this is actually very old methodology. But here's a mature Platycerium hilii gametophyte that's about 10 weeks old. And I photographed this using a stereo microscope at about 10x. And just barely emerging from this gametophyte is what's called the sporophyte. This is the diploid plant that will grow into the mature plant that we would see hanging up in a tree somewhere in the tropics. So what on earth did I do to become worthy for a degree? What I learned in my research, the Staghorn gametophytes are what's called protandrous, which means they develop Antheridia first and they develop Archegonia next. And this, from a propagation standpoint, this is very important, because if the gametophytes are going through a male cycle for a few weeks and then they switch and become female, you're not going to get a lot of fertilization, and therefore your propagation yield is going to be really low. So what I discovered is I could enhance the production of Staghorn ferns by overlapping the sowing. So production of plantlets is enhanced by creating an overlap of gametophyte maturities in the culture, where I can guarantee there will be Antheridia and Archegonia present at the same time. So I very simply applied a second dusting of spores about a month after the first one. And this created a diversity of age of the gametophytes in the culture, similar to what would happen in nature. The other thing I learned is I had to manage the water level in the culture, because again, when you think about favorable environments in nature, moist mosses or tree bark are very favorable to these plants in terms of their reproduction in nature, but they cannot be submerged. The other problem I had in the research was the cultures were very sensitive to infection by blue-green algae. So, another propagation tip I would offer is try to keep all of the utensils and culture media that you're using clean. It's probably not a good idea to put it in the oven to sterilize it unless you're just using sand. But I would use boiled rainwater to make up your culture media, and certainly use very clean containers. Maybe run them through the microwave before you begin to assemble your fern culture. And then again, I used a fine artist brush, which I sterilized in alcohol to dust the spores. And again, this keeps them from being too crowded in the culture. So what do the cultures look like after the gametophytes are actually growing? This is actually from a research study that's fairly recent, I think in 2010, but you can see here the researcher was using glass jars, with paper over the mouth of the jar. In this case, this researcher did not use what I call the paintbrush method, and you can see the gametophytes here are very crowded. In this particular approach, the researcher actually lifted out pieces of these complicated mats and put them in pots. And then you can see here the diploid or sporophyte plantlets are hatching out from the little pots here. So any clear container that will hold moisture will work. All the culture materials, again, need to be sterilized, and the staghorn plantlets emerge about 2 months from the sowing of the spores in the culture vessels. I thought this picture was interesting because it shows the staghorn ferns reproducing on moss. And again, this is what you would typically see in nature. These are Platycerium madagascariense plantlets, which, if you're interested in collecting these plants, you can actually buy these from propagators, and they're fairly easy to grow. This moss is actually growing on what looks like osmunda, which is very easily acquired from orchid supply, or I've seen it in some nurseries. So there's only 18 species of staghorns in nature, and they typically get divided by geographic region. I've got several of these in my own collection. Some of these are very difficult to grow, and a few of these Malayan Asiatic species, like Ridleyi, Platycerium wandai, for example, and Wallichii, they're single bud plants. And when I was growing these in Florida, I had a lot of trouble with them being attacked by pill bugs. A lot of the Javan, Australian, and African American plants are what I would call rhizome branching. So they produce, they produce a group of plants on the culture media, and they're much more resistant in many respects to the attack by different insects. These are all mature staghorns that you see here. Platycerium and Dynam is from South America. This is actually the only species that's native to the Americas. And you can see the morphology of this plant. It's growing up in the trees, and therefore it would be classified as an epiphyte, similar to Spanish moss, for example. All staghorns produce two kinds of fronds. These, what many people call the crown fronds or basket fronds, actually collect detritus falling from the trees, leaf litter, and it sort of forms its own soil, against the tree. And then these are the fertile fronds that produce the spore patches where the spores are actually produced. Platycerium angolense is from Africa. Haltumii, which I actually have in my collection here in Weaverville. You can see some of these things look almost like they're from outer space. They're very bizarre looking, both in culture and in nature. Platycerium bifurcatum is the species that you'll buy if you go to Lowe's or BB Barnes. This one is actually quite common in horticulture. And then here's a couple of pictures of my own collection. This is actually our house in Florida, where I had these hanging on a wall. And this is a Platycerium haltumii that I mentioned to you that seems to be very happy outside. If you're interested in fern resources, there's a couple I recommend. The Fern Growers Manual, which is by Dr. Hoshizaki and Robin Moran. Barbara Jo Hoshizaki was one of my advisors on my graduate school research, and she published this great book, which you can actually buy online. And then Roy Vail is sort of a longtime historian of Platycerium culture by hobbyists, and he published a great handbook for anyone interested in these interesting plants. Here's some more resources for you that I wanted to list for you if you care to go look at their websites. The American Fern Society Partners website has a lot of resources. American Fern Journal, the American Fern Society has a separate website, and then there's actually a collection of fern websites that's been curated at this particular link here. And then the Los Angeles International Fern Society was a substantial contributor to me when I was in grad school. And here's a couple of others that are offshore. So in conclusion, as I say, once upon a time in a galaxy far, far away, I was growing staghorns and was able to discover how to propagate them in the laboratory, which now, of course, is fairly common in horticulture. And, I really want to thank my major prof, Hank Wayland. He's in his 80s now. He's actually well known in the city, his retirement town of San Antonio, Texas. B. Olson from LA International Fern Society, And sadly, I had to celebrate the life of Dr. Hoshizaki. She was one of the very first researchers in the world who actually took a hard look at these plants. And the Botanic Gardens of Berlin, Brooklyn, Kew in London, Missouri, Philippines, these are the institutions that supplied research material to me. And I've been able to visit a number of these over the years, and it's always great to see what they have in their collections. And I want to thank you again for inviting me, Warren. And
Okay.
I'm certainly open for questions.
So my first question would be, how are you keeping things in Weaverville? What kind of circumstances do you have your plants in?
So Jim, the house that we bought here had a very big deck, and we actually covered it last year. And part of the deck, I created a solarium or a plant room. It has lights and windows on two sides. And this winter was my experiment in terms of how to keep it warm. And I bought the windows for Window World, and our carpenter insulated the walls very heavily without any heaters or any kind of supplement. It's typically 20 to 30 degrees warmer than ambient. Even when it was 22 degrees here, the other day it was 62 degrees in the plant room.
Amazing. You must not have too much shade around it.
No, it's facing west. So, one thing I've discovered, and again, I'm still in discovery mode with this space, but the western sun comes through really bright, and some of the ferns that I had against the windows that were actually burning, and I had to move them away.
Do you have to keep the humidity high?
I don't, because my wife runs the clothes dryer almost every day. And so I have vented our clothes dryer into the space, and it really dumps a lot of humidity in there.
Creative, very creative.
I'd like to ask Phil, my interest in plants is mostly going to be as far as hardy ones outdoors, and we've had some native ferns as well as ferns we bought. Would if I wanted to propagate, I know I can divide a number of them, but would I also be wanting to do the same process of collecting spores, etc., to do sexual propagation of hardy ferns?
Well, here's what I noticed, and I have a number of osmunda pots, in my equipment. If you create a space either outdoors or inside, wherever you're managing these things, that's favorable to the gametophytes, you'll get volunteers everywhere. I had bird's nest ferns that I was giving away to the neighbors, often that would just pop out, on some of these osmunda pots that I have.
Okay. Well, we have some cinnamon ferns that have been spreading like crazy outdoors, and I'm very pleased about that. And Japanese painted ferns, they do wonderful. I just didn't know, I can divide them, but I just didn't know, would it be sort of a fun, or would I get much different by doing sexual propagation?
Yeah, my suggestion would be, certainly take advantage of the volunteers in your garden, but if there's something you really like, you might want to try this method of using a dish. I know I see Glenn is on with us. Glenn has a beautiful moss garden. And, we might want to ask him if he actually has fern volunteers in his moss garden. Other questions?
I bring my Boston ferns in the late fall, and I store them in the basement under some grow lights. I heard Tom mention that he has ferns outside, cinnamon ferns. Can I leave my Boston ferns out over
No.
winter?
No. No, they will freeze. The natives that you saw in the list are very hardy, and they actually may die back, but they'll usually come back. I mean, for example, holly ferns in the garden here, I notice the leaves are all burned from the cold, but they always come back.
Okay.
Yeah, there's several really hardy native ferns, but Boston fern is a tropical that's been brought into the horticulture industry actually more than a century ago, and it's probably the most popular commercial plant because it's so easy to grow apparently, and cheap, so you can always buy one for practically nothing. But they won't survive outside.
Thank you.
What are the ferns that seem to grow wild in Western North Carolina that look like Boston ferns?
Well, I have in my yard, some of which are native and some of which are things I've brought in. I have maybe 10 different kinds, but the hastened fern is a native fern that dies all the way back, and it's really quite a nice fern.
Okay.
But I have others that also fit in that category. I don't know, Phil, do you have some comments on that? I didn't mean to take the floor.
No, I again, I think, a number of the nurseries actually, I know here in Weaverville, Reams Creek has a collection of native ferns, which, if you go out there now, you can see what has survived the winter, in their lath house, and, what's died back. But, I think, planting natives is really a good rule of thumb, not only for their survival, but I think they look great in the garden.
The one that sort of stays evergreen here is called the Christmas fern, and it's quite abundant. And most of mine were just native underneath the in the back. You can see behind me here. You can see a fern over here on my right here. That's a sensitive fern. No, excuse me, that's a hastened fern. I have sensitive ferns. I have hastened. I have Tennessee glade ferns, all of which come back. It's not any problem. But they'll all but the Christmas ferns freeze all the way to the ground and come back prolifically.
Yeah.
And they also seed volunteers in my mossy areas that are getting worse with this extra rain we've had. And so they're just coming up. And I assume they're following the propagation sequence that you outlined.
Yeah, it's certainly true. I remember when I actually did a project for the Corps of Engineers a long time ago, and found a habitat. This is when we lived in Texas. I walked down into a creek bed that was probably 20 feet below grade, and the entire channel of the creek was lined with maidenhair ferns. And, I expected to find those here as well, growing wild, but I haven't found them yet.
I've got lots of them on my property.
Oh, really? Okay.
Many clumps, square feet in area.
Yeah, so
I could donate some.
Yeah, so those I think are also very attractive because the fronds are really delicate, and there's a really nice ground cover feature to them as well.
One of the places I've gotten ferns is when we had the plant sale out at the botanical gardens. Some of the native plant vendors have ferns to sell there. They're, I bought it and transplanted into my scheme. But I also know that I dig quite a few, and I can share substantially.
Bill, I'm wondering if the staghorns are considered endangered or protected in any of the nations that where they're native.
Yeah, well, that's a very good point, and they absolutely are. I mean, that 2010 research that I was showing you, the fellow was at a university in Brazil, and everywhere humans are deforesting the jungle, Madagascar, Angola, Brazil, Vietnam, the habitat for these things is disappearing. But I have to say the other, we were at the Marie Selby Botanic Garden, the last visit, and I was pointing out in the trees all around the Botanic Garden, there are staghorns everywhere. So, there are parts of Florida where they seem to be reproducing as they would in nature.
I've had staghorn ferns, and I guess I'd never realized what the shield frond is. And it seems I think initially it was sort of greenish, but it's turned brown and dried. I guess it's dead. Is that something you should remove, or?
No, Mike, again, in nature, the shield frond forms sort of a basket to, collect leaf detritus that's falling from the host tree. And, in some species, for example, Platycerium ridleyi, it creates this symbiosis with an ant colony, and the ants will actually protect the plant from being attacked by something that will eat it. So, when I mount them, I usually buy sphagnum moss and, get it wet, and then, put the staghorn on the moss, and then I tie the plant down to the moss, and you'll eventually see that the fern will begin to produce the root system out onto, if you're putting it on a board or, in some kind of basket. But no, I mean, to answer you, I would let those fronds, stay where they are.
Okay.
Okay.
Bill, thank you much. I gotta go, so I'm gonna ring off, but I thank you so much.
Jerry, good to see you.
Take care now.
Take care, sir.
Yep, okay, thank you.
Thanks a lot.
Thanks, Phil.
Thanks, Jen.